to filter into the room. So hang on. Again, welcome everybody. We're just giving it another moment for people to filter into the room. Welcome everybody. Let's just give it maybe another 10 or 15 seconds for people to filter in the room. Okay. Again, welcome everybody to the NIIMBL webinar series. Today we're going to have a webinar on end-to-end automated Glycan Analytics platform for model-based adaptive control, a key use case of NIIMBL Big Data program. And it's going to be given by Abhijit Sawikar. Before we get started, there are a few announcements. So throughout the presentation, everybody's going to be muted. Please use the chat feature to post any comments or questions. All comments and questions will be held to the end of the webinar. And please keep in mind that this webinar is open to both NIIMBL members as well as non-members. Please do not post any NIIMBL confidential or proprietary information in the chat, and also at the end of this webinar, attendees will receive a survey link. So please fill out the survey. We value the information that we get from the survey, and once the survey is submitted to NIIMBL, you will gain access to the webinar recording from today. Next slide, Abhijit. Thank you. So we just want to bring everybody's attention to we have a big data program workshop coming up in March, the 15th and 16th. This is open to both NIIMBL members as well as non-NIIMBL members. Day 1 will be open to everybody, and then day 16 will be for NIIMBL members. There is a live in-person portion of this as well as a virtual portion, and the in-person portion is in Newark, Delaware. So please, if you're interested, please register. You can register on our website. You can go to the NIIMBL website and navigate to the registration form. If at all possible, it would be great to have people in person in Newark. So please consider coming in person, but if you can't, please join us virtually. And to just, if you're interested in participating in the Big Data program and or Big Data Workstream 3, please reach out to me. My email is jjsaylor@udel.edu. And then if you are interested in joining NIIMBL, please reach out to me again at jjsaylor@udel.edu, and I can put you in contact with the right NIIMBL representatives. So at this time, I'd like to introduce Abhijit Sawikar. He is an innovation manager within the Global Analytical Pharmaceutical Sciences and Innovation Organization of Global Analytical Development, which is within global CMC development of the healthcare business of Merck KGaA in Darmstadt, Germany, operating as EMD Serono in the US and Canada. Abhijit holds a core background in biotechnology with scientific expertise in large molecule modalities, both marketed and NBEs. He has a core expertise in analytical development with subject matter expertise in glycosylation-related topics and is a member of the Site Digital Committee as the responsible leader for selective digital projects. In his current role, Abhijit manages innovation activities in the area of biotechnology state-of-the-art analytics, QC automation, and digital innovations in both GXP and non-GXP environments. Additionally, Abhijit is actively engaged with business schools as an external supervisor to PhD management programs within the theme of digital transformation. And with that, I'd like to hand it off to Abhijit.
Thanks a lot, Jim, and really a nice opportunity to also present on behalf of NIIMBL the work that we have done in this BD3 stream. So before to start, I would want to point out to a disclaimer here that I am a part of Merck Serono SPA based in Rome, which is a business of Merck KGaA, Darmstadt, Germany, and operates as EMD Serono in US and Canada. So all the views, analysis, and the perspectives are fully independent and belongs to the work performed in NIIMBL Big Data program, which is together with the participating companies and organizations. So they do not represent the views or opinions of Merck Serono SPA, which is again the business of Merck KGaA Darmstadt, Germany, operating as EMD Serono in US and Canada, or to any other company or organization. So this presentation is intended for informational purposes only, and it is not intended to promote any products or services. So with this, let's dive into this webinar on this topic of end-to-end automated Glycan Analytical platform for model-based adaptive control, which is a key use case of NIIMBL Big Data program. So just to give you a background on the Big Data program, which was also provided by Roger Hart in his webinar on 16th November. So Big Data program is been established at NIIMBL, having these 5 key streams. So the first stream, which is working on standardization and contextualization of the data. Then the second stream is working on defining the process control at the NIIMBL Test Bed. Then we have the third stream, which is my stream, that is providing support on the multivariate analytics and sensors that are needed for the Big Data program. Then we have the fourth stream, which is about generating the rich data required as a training set for the models. And then the fifth stream, which will be the one that is currently working for creating those models, those simulations, and so on. So of course, coming in the future, each of those streams will be providing you a bit more background on the work and the outcome that has been doing in their part in those streams. Currently, in this webinar, we will focus on the BD3 stream, and the objectives of our stream has been to develop and test at-line, in-line, or online glycosylation or glucose sensors for its use within the setup that the BD2 has been establishing at the NIIMBL Test Bed, and to develop multivariate sensors to cover CQAs. So talking about amino acid detection, cell viability, and so on. So overall, our goal has been to reduce the need for the QC laboratories when we talk about smart manufacturing, and bring let's say the offline analytics into inline or an at-line measurements. So overall, objectives that have been pursued within this Big Data program has been to facilitate data exchange and transfer through the creation of meta libraries, ontologies for cloud-based data transfer. Then develop and demonstrate the adaptive process control of CQAs utilizing multivariate sensors and process analytical technologies, and to have the use of advanced process models to improve process understanding, to enable speed and reduce experiment demand. So we will be focusing more on this second part, that how we are supporting the BD2 stream in order to set up those analytical setup that is needed for the adaptive control. So the Big Data program is really an enabling program that will support the needs for future biopharmaceutical manufacturing as well as the other NIIMBL initiatives. Okay, so going a bit more granular here. So the Big Data program work streams have their defined objectives from moving towards data creation, data transformation, data storage, prediction from data, and control from data. When we talk about data creation, Here, the key use case that we are pursuing within the NIIMBL Big Data program is on the NIST and within the current scope, we are going to demonstrate the glycosylation adaptive control in the upstream process step. For this, there is an instrumentation that has been set at the NIIMBL Test Bed along with the sensors and so on. So this would this would be the basis of the data creation that would happen at the NIIMBL Test Bed. Then our part comes in, which deals with the data transformation. And as I mentioned earlier, that about our objectives is to define these sensors, which could be used for this inline sampling and derive, the information with advanced analytics to facilitate the controls of sequence. So this program has been ongoing from the last year, where last year we have, worked together to develop this capability for this at-line glycan measurements at the NIIMBL Test Bed. We have also extensively worked to prioritize the opportunities where multivariate sensors and associated data analytics can be applied within this big data context. And as we move into the 2023, so we want to demonstrate the at-line analytical platform for glycan measurements and then define what could be potential work that we have to initiate in order to bring this high value analytics for other opportunities. So coming on the next slide here, next block here, which is talking about how we are going to set up the data storage, the prediction from data and the control from data. So I would very high level go through it, that we are defining the setup at the NIIMBL Test Bed, which is driven by the BD2, having a sampling device, a liquid handler, which would also follow a typical IQ, OQ, PQ procedures in order to make it quite industry-oriented type of setup. Then there is a BD1 stream who is also working on developing this process ontologies, which I guess means this information would be available to you in the subsequent seminars that we will have next week there. So with this means, let me move to the next slide here, which talks about who has been our participating organizations and the contributions to the outcome that I'm going to present. And I'm really thankful for everyone's contribution here. So we have quite a nice share of industry representatives, all coming from the big pharma companies, as well as vendors, universities, and the national bodies such as NIST and so on. So again, means thanking them for their contribution in the outcome. Within the big data stream, we are organized into 3 substreams. So the first stream is focused on support and giving the support to the NIIMBL Test Bed on developing the workflow and the capability for the automated at-line glycan analysis. And going further in this webinar, so I will be only detailing aspects related to this substream. Then we have a second substream, which is working on analytics, which works as an analytical prioritization team to evaluate various novel sensors and technologies to control CQAs. So this is where means when we want to step further and look for CQAs that are beyond glycosylation and tap those opportunities. And finally, the third stream, which is really about data, which means that how do we bring the data that is the signal to the attribute information. So currently, we have a lot of emerging trends that are happening around where multivariate techniques have been applied, and that is where this team is going to look for opportunities and then prioritize those areas to be brought in for the further extending the big data program. Okay, so we are now moving into the work as an outcome that was driven in this first substream on the glycosylation setup. So starting with the URS, which was designed in discussions with all the participating work streams, our aim for the BD3 here is to set up an at-line sampling, to set up the sample preparation that will be needed, to define which sensors would be best suited for this purpose, and finally to play with the data, means for the data processing and to make this data in a digestible format or I would say a ready to use format for the models in order to give the predictive outcome from this. So, the decision making for a change in glycosylation at a bioreactor level. So this usually happens within 6 to 8 hours. And that's why this has been our, let's say, the prime guideline that our analytics has to meet within this timeline. And we started to explore a bit more in defining what should be the at-line sampler based on the specifications, the tubing size, ports, the sampling volumes that are needed. also looking at which sample preparation could be best suited to bring it as an at-line mode. So, there are various sample prep approaches which range from automated to manual, so from pep map to glycan mapping assays, and also to think about how those assays that we are going to select are also going to fit within the titer levels that we are generally going to see within this process. Okay, so just to add up, the process that the BD2 is going to demonstrate at the NIIMBL Test Bed is going to be fed batch process for a deep classical map. And so we are expecting some changes that are going to happen over the days as per the titer there. Then moving to the sensors. So, currently there are a lot of technologies that are available, which range from spectroscopic sensors to mass spec sensors, which either use the front-end separations with chromatographies, capillary electrophoresis, and so on. And within this last block means we also have assessed what are the various data formats, how immediate once the data has been acquired can be processed with those softwares that are available. Are there any conversions that are needed to move the data from, once it is acquired, processed into the model ingestion part of it? So all of these factors, they have raised a lot of discussions across the team, and we started to build this up as an analytical target profile for our use case, and typically following the AQBD approach, which is analytical quality by design. Now, the second step was to select the technology. And here we have applied a systematic approach. So starting first with the concept evaluation. So based on the URS, as we discussed before, we focused on speed. We focused on having it fit for purpose and in scope for the process monitoring. So for having a technology scouting, so we have chosen all the, let's say, the major glycan analytics techniques that have been widely used by industries, from released glycans, LCMS MAM, LCMS intact subunit, multi-MS, capillary electrophoresis, as well as the plate assays. And what we have designed is a rubric here, which would allow a faster decision making across these various domains in terms of performance of those analytics. So typical, SLAP approach, where we assess, the specificity, linearity, accuracy, precision, if it is really meeting those needs for the measurements. The technicality, that is the readiness around the readiness of the technology to be easily applied into the biomanufacturing space, as well as risks, means the stability of the tech, the fluidity of customizability, as well as the connectivity with the other systems and alignment to the URS. And finally, also the aspects about the business value, where we have just looked at qualitative aspects of it, means if it's really going to fit on financial side, non-financial side, strategy, even at the level of sustainability and so on. So just a very quick input on how the scores mean. So we have conducted a sort of a typical survey, decision assessment using scores of 5, 3, and 1 within the team, and came up to this conclusion. So as a result of this, so the first method of choice has been the LCMS intact subunit, and the second method of choice has been the LCMS MAM, which has been always aligned to the trend that we are seeing outside in the literature. Now, what is this LCMS intact subunit analysis? So this author means they have really given a very good background to our selection as a prior knowledge, I would call it, where the middle up MS approach, which is a very simple setup, it generally needs your MAB, digestion with an enzyme to digestion at a hinge region, and then the use of reductant to reduce it, so that it also provides a homogeneous preparation there. And with an helic MS approach, you can even very nicely separate it chromatographically. And what we see here from the data, that all of the major species that can be observed with the classical 2AB glycan released method are also detected with the middle up approach by using both fluorescence detection with UPLC as well as the MS signal detection. So, this really gave a very strong confidence that the approach chosen has been correct in terms of moving ahead for the decision that we need for the process. Now, going a bit more in depth about the analytical quality by design approach that we have been following. So we have the analytical target profile that has been set up following the process and the business expectations. So in this case, the program expectations for the within this Big Data program. The technology selection has been performed with having say having these assumptions and the evaluation based on its technicality and also on its practicality to align and meet these expectations. This was provided, thanks to all the experts that were participating, to the vendors that were also even quite open to provide the details on the technologies. From the prior knowledge, the platform method has been selected as the middle up LCM match, where we do have preliminary results from the NIIMBL Test Bed, where we are able to deliver the results within 4 to 6 hours. And what you see here is really a beautiful profile. So you clearly see major species of the glycans that are needed at a process control level with an LC as well as the MS. What we are going to do coming in the future, that we are going to see the adaptations at an at-line if there is some adjustments that are going to be needed, and also to define the comparability to the offline analytics. So in this case, the gold standards that would be chosen and so on. We are going to also have the method performance evaluation to demonstrate it as a fit for purpose, to define this method operating ranges, definition of these SSTs if it is needed, and so on. And finally, as we are going to deploy into the routine and see its monitoring and evaluate if it is meeting our expectations. So again, I would call it more or less like a first generation of analytics that would start with, and there would always be an opportunity to see and to bring in the second generation, the third generation, as and when the technology evolution happens across this industry. Now, this slide here, which it shows you a bit more on the setup that we are going to have at the NIIMBL Test Bed, which is going to bring this end-to-end analytical setup for glycans. So what you see here, that we have a bioreactor that will have its process parameters and raw materials input, which would be controlled with a controller with the set points, a classical setup for a biomanufacturing. We are going to support on setting the sampling device, the liquid handler, the LC system, and the mass spec, which of course have been provided by the different vendors at here around, and the outcome of this would be used into the models, which would then define the adaptive control needed for the controller to adjust the set points in needed. Now, what we are also looking currently is that we have these different modules, and there we see, let's say, a challenging need in order to harmonize or connect all of these modules together in order to have seamless connectivity across these platforms and drivers, in order to also have the automation of all of these analytical equipments with controls, with data processing, and so on. And also to within our stream that we have been working on the signal to attribute, that how we could automatically translate the univariate or the multivariate signals into attribute information for control. And then the topic of standardization comes in, where analytical data ontologies would be pursued in order to harmonize and standardize the whole structure. So here there is an emerging need that we should look into these aspects of focusing on this analytical operational technology and IT core in order to address some of these challenges that we are currently facing to have this end-to-end analytical setup, and which again opens this opportunity to the community here, where they could come forward and then let's discuss about some solutions that might be suited. Now, as a path forward, so this is again taken from a webinar given by Jean Schoffer, which was about the introduction to NIIMBL on the 7th of December. And what was communicated here, that we have this introduction to NIIMBL use case that NIIMBL has produced. Here, what we see that there are a lot of analytics that are currently provided as an offline there. And our aim here, when we are going to pursue the big data program in future, is to also align and prioritize these CQA testing needs with the emerging innovative analytical platforms and approaches that we would come across, and make sure that we are making this transition of this offline analytics into the inline, online, or at-line setup. And also, again, as I mentioned before, that to bring this multivariate signal to attribute approaches here, which is also an emerging trend happening across the industry. Then also there is another interesting aspect, that how do we shape the analytical strategy in the context of big data. So this again opens a lot of arguments that we need to emphasize, or we need to reflect on, and that is where we want to start a thought process here. So when a molecule, it moves from your phases, from your early developments to clinical manufacturing, registration, and into commercial. So there is an analytical strategy that has been, already been established, or many of the companies are following it. So we want to reflect means how this analytical strategy should be adapted within the scope of big data. So define what phase appropriate means in terms of the analytical platforms, in terms of this analytical data generation, and how they could be used to support interpretations for the controls. So that's where means we want to dive a bit deeper and then start a thought process there. Then there is another good topic which is about analytical data quality. So the data that is going to be generated by different analytics per se, let's say, because we have to tap on speed, we have to tap on, let's say, information that is phase appropriate. And if we are using different analytics, how the interoperability has to be defined. And if those data can be categorized as fit for intended use within big data. So, such aspects we really want to touch on. Then there is there is also an open area on the testing strategy. How we have to make it coherent in order to support the process decision making. So we are acquiring the data which is really going to be used for a process decision making. And as we move ahead for the future, so we want to also think how do we adapt a typical analytical lifecycle management approach here. So in order to bring these continuous improvements to deliver robust and reliable methods in the context of the big data program. Now, this slide here, it shows you a typical panel, what is being used as a quality attributes for a MAP DS. So currently we are focusing on glycans, where the current approach as an offline has widely been used by industry with release glycans. And here we are trying to tap the middle up LCMS at the USP as an at-line method, which is currently in progress. What you see here, there is an open ground for an opportunity to contribute as we move further with to tap the other CQAs here, from chart variants to aggregates, fragments, PTMs, and so on. With your domain expertise, and if you have also some solutions which you think would be tapered at certain unit operations within the biomanufacturing. So that's really an open opportunity. And so now talking a bit more in context about what opportunities we have in the frame of big data program within the BD3 stream in itself. So as I have highlighted to you that we are really focused to bring the state-of-art analytical technologies that are from the emerging innovative sensors, multivariate platforms that are available. Then we are quite focused to enable the end-to-end automation. So starting from sampling to sample preparation and to data architecture of the analytical platform, and to demonstrate its practicality at the NIIMBL Test Bed. Then, as we have seen the last slide, which lists out the quality attribute panels. So where we are also going to prioritize which are the industry's need in terms of difficult to measure CQAs for bringing the adaptive control into manufacturing. And we are also open to listen the challenges and the gaps that you have, where you see are some of, let's say, the roadblocks, I would say, for a smart manufacturing. So you could even come up with such topics, and we can look into these directions, how best we can see for this industry's need. So coming to this slide means I want to thank a couple of folks here, so which is starting from the big data program work streams. So BD1, BD2, BD3, BD4, and BD5 member participants. I want to thank the big data steering committee for consistent guidance during these review meetings and so on. The NIIMBL members organizations who have been contributing to this big data program, as well as our external contractors and collaborators who have been contributed to the outcome of this work. And then to the organization of the NIIMBL Big Data program, which has been led by Roger Hart as a program lead, Jim Slater as a program manager, Sandeep Kedia as a program support, and Taylor Zolat as a program support here. So with this, maybe I would hand over to the NIIMBL representatives for some questions if we have.
Thank you, Abhijit. Yes, we've been getting many questions in the chat, and I would remind folks to please enter your questions in the chat so we can ask them accordingly. Pertaining to your presentation, there was a question around slide 8. Is there an intent to apply the same setup the state of the art glycan analysis in a continuous process at some point in time? I know you described a batch reactor or a fed batch reactor, but can the same strategy be applied for a continuous processing?
So I would say that the analytics would be quite general in nature. So since if you know we are aligned on how what is the sampling, what is the sample preparation, it could easily be extended to any setup. So even you know the perfusion mode or a continuous manufacturing mode.
Pertaining to that question, there's a follow-up question. Can you describe the objectives and the need to what level and end-to-end control is desired in manufacturing environment?
This is a great question. So I would say means we are still at an explorative phase here, and we are currently looking at a glycosylation as a first use case. So you know our work has been focused only on the glycosylation CQA as a first use case here. Now, okay, so there would be also the other streams who are working mostly on the modeling, which will be much, you know, they would have a much better explanation to this question on how the data that is going to come out from the analytics would be used in the modeling part of it, and which could, you know, be further extended for an adaptive control and derive, you know, and change, you know, manipulate the set points to keep the product quality consistent.
Okay. There's another question around some commercial solutions available. So can you elaborate a bit more on the choice selection for the data integration, processing, multivariate analytics challenges that you mentioned in your during your presentation, and the solution that may already exist commercially on how they can enable you to resolve those challenges?
Right. So, so in this case, let's focus on the current use case, which is about the glycosylation, where the signals that would probably be acquired by an LC system or a mass spec system, right? So we know that LC system processing is pretty straightforward. So we have well-established vendors who are providing the peak integration process. But again, the peak drifts, the retention drifts are again a challenge. This is where means we may need certain upgradation to the existing solutions that we have on the LC signal processing. On the mass spec side, I would say that there is a lot of variability here in terms of instrumentation, in terms of, let's say, the different principles on which these instruments are operating on the mass spec. And that also, you know, brings into this discussion the different file formats that would be generated by these different instrument types. So this is where means we also want to narrowly understand means how interoperability can be defined when we move from one mass spec to another, and if there is some factor that we could work around. And you know this is a topic that our stream with signal to attribute would take into hand. So how the deconvolution has to be set in place. Are there automated methods that can do this and that can perform it? I can tell you that there are also, let's say, third-party vendors that are capable to process the data that is coming from wide variety of mass specs, but just not naming it here just because for the sake of you know confidentiality and so on. But if you can browse around, means you could find some solutions that would satisfy this need of the signal processing here.
Thank you. There's a question on the strategy, the overall strategy. So can you elaborate a bit more on the strategy used to down select technologies such as for Glycan, or what could be potentially used for development versus commercial aspect?
Right. So, you know, as a typical analytical strategy, what we use, right? Of course, when you are defining the screening for your DOE experiments and so on, so you really need fast and you really need something that is really going to give you an indication. So depending on what is the scope of your, let's say, experimentations, you could either choose your analytics. So in this case, a plate assay would be give you the relevant performance there. But since we are going to talk, or let's say, since we are going to use this data for a big data program, so we wanted to have more information than the plate assay and less information than the released glycans. So released glycans methods are basically used for characterization purpose. You know, this is well established, right? So there is no doubt about it. There are technical challenges when you have to bring it as an at-line method. So with respect to the speed, the fit for purpose, motive, and so on. Within the process, since the aim has been to look at the major species in order to control, and we wanted to choose a method that is capable enough to give us, let's say, the quantity or let's say semi-quantitative estimation of the major species in order to drive the adaptive control. Now, talking about, these aspects, like how feasible or how easy the method would be to be set as an at-line. So we don't want to give, let's say, our manufacturing guys too much of an effort in order to, cook those samples when we talk about a peptide mapping or a released glycan. There are multiple steps involved, multiple variables involved, and this is what we wanted to minimize at the first time, right? And that's where the intact subunit approach, it really fits in. And there are also some articles that have been published by Genentech, for example, who has used the 3D LC approach in order to use this intact, let's say, subunit LCMS to be used for a process monitoring and so on. So those has been the prime factors where we narrowed down and to choose which is really the simplest one. Of course, looking also at the business value that it is going to generate, and that's why you see it scoring a bit more higher in this categories here. And then, of course, you have to also think that this type of selection has been taken in considering a typical classical map. If we are talking about multiple sites of glycosylation, of course, the subunit may or may not work. And that's where you have to really go for this LCMS MAM, which it's going to provide you site-specific distribution and to give you much more better detailed information. So that's how the overall strategy and the results looks like with this assessment.
Thank you. I think there's a question around the selection for the glycan. So glycosylation is a great use case, but also a very highly complex CQA.
Right.
Subject of such scrutiny by regulators. So have you considered regulatory receptions of these approaches? For example, very few MAM methods have been approved.
This is a great question, and I think this also brings into this discussion about this establishment of MAM, right? So in the consortium, there has been an effort to establish this LCMS MAM as a method, and in order to adapt, more or less, I would also call it more like a conservative strategy, thinking from a quality background and the regulatory point that you highlighted. We are going to use these two sensors, which are equally going to deliver data that is, going to be consistent. So for example, the fluorescence detector by an LC approach, so just peak integration and the outcome is there. So the LC approaches are well established. So there are pharmacopoeial guidelines in order for them to be, regulatory, in order to have them regulatory accepted. And then the MS approach, which is emerging. So, so the strategy that we are thinking now is to use primarily the LC approach in order to get this setup accepted, and to use the MS as a secondary sensor. More or less, you can even call it like an investigational purpose and so on. But let's see how the outcome and the result looks like when we will move into the qualification exercise, and see how interchangeably we could use either of the both sensors, or if just one LC approach is enough for a classical map.
Thank you. There's a question around your strategy again, around the glycan measurement requirement for adaptive control, which was the 6 to 8 hours described earlier. What are your thought process around more complex glycosylated molecules, especially with the ones that have multiple sites? What is the impact of that on glycosylation, or how would you go about setting that up for adaptive control?
So this is a great question, and I would relate again that the LCMS subunit is more suited for a typical classical map. If we end up into more complex formats, so I think without doubt the LCMS MAM would be the best choice, because that is where you would have the distribution of the glycans, which can be given by the peptide populations, and you will have the different distributions that the technique would deliver. Now, with respect to the adaptive control, I think there needs to be some knowledge and experience that needs to be gathered before defining what to control and how to control. And there needs to be these Dewey experiments, which will tell us which are the perturbations that are giving rise to the changes. So I would purely relate this based on an experience-based exploration approach, how the adaptive control to be defined on such complex formats.
Okay, thank you. And this is one related to the sample prep. in terms of how do you integrate peptide mapping in a PAT tool when the processing time with respect to trypsin digestion is extensive? So I think this is a method of choice and related to method prep, sample prep.
Right, right. So yes, so there are certain variants that are also available in terms of rapid digestion kits and so on. So such approaches can be used in order to bring it at line. Again, consider that the use case, let's say the turnaround time that we need for the glycosylation control is 6 to 8 hours, and this is still doable in order to have a peptide mapping sample prep there.
And I do believe there was a question around, will these slides be available? So I'll answer that question. Yes, as soon as you fill out the survey, we will make sure the presentation is available to you. The slide deck won't be, but the presentation would be. Please add in the chat any questions you may have. There's another one around the NIST map is well defined by now. How do you create variants to understand what detection methods can be accomplished?
I don't know, Sandeep, are we, can we answer this question based on confidentiality, or?
I think we can take it offline. So whoever asked that question, we can kind of, if you can just email Jim and we can kind of get back to you on that one. So there's a question around GMP use. Do you envision this? Sorry, do you envision the technology can be used in GMP, or do you imagine primarily to be in development? So I think that was a question around development and commercial. So you can elaborate that, like how would we make this in a GMP environment?
So at least means we want to start with the development environment for the big data program, and again I will relate this to the experience-based exploration approach, and then start to move, finalize and let's say lock down such platforms for the GMP use. And currently there are technologies which are evolving faster and adhering quite a bit towards the GXP regulations and so on. And I believe health authorities are also open for such emerging, innovative analytics and so on. So for sure means we would need a joint effort together with suggestions and recommendations that the authorities would provide once we provide this experience-based exploration data on such innovative analytics.
Could you describe a little bit more information around the future CQA that you mentioned that you would like to tackle on? What would be the ask and how would you go about developing those?
So, currently we are defining the prioritization of all of these CQAs. And I believe this is also a topic that the NIIMBL organization has to also take in terms of, how the program would be extended and so on. So, but I would just talk about more on the technicalities of this decision, which would be, of course, driven by the industry needs. So I believe that Sandeep, if you want, means we can highlight that there would be a probable survey that might come. So we are still in discussions how we are going to administer this survey. Either it is going to be just within the NIIMBL membership or also including non-members. So this would come as a next step, and this would bring the needs from you, from the community to us, and then start defining which would be the next CQAs, and following that means we would again indulge in listening to various platforms that are available, which are best suited, and to go forward. Of course, there is a lot of information that is available in the literature, and we want to make best use of it as a prior knowledge, how we have shown in this case as well, right?
Yeah, I think the question that they might be asking is how do they get involved? So I would say, please feel free to reach out to Jim. It was not so much about prioritization of these CQAs, but more about how they would get involved. And from the ask from this work streams specifically would be, what kind of development are you anticipating? You've gone through extensive development for the Glycan Analytics. Would you follow a similar pathway for the other CQAs as well?
Okay, so coming from an industry perspective, I would say yes, because since this is an opportunity that the NIIMBL Test Bed is providing us, right? So in order to really go there and test the practicality of those approaches, and not just theoretical assumptions and concepts and so on, and that is where means as an industry representatives, I would really vote for this one.
There's another question around the upstream process step. So which role of UPS development has in this work stream to see which conditions affects glycosylation? So this is about modulation for glycosylation.
So currently, I believe means we are playing around with basic process parameters. So typical temperature and so on, which is also well established in the literature, that they are responsible for changing certain glycosylation distribution and so on. For example, high mannose with the temperature shifts means you can play around with that, as well as having some feed concentrations, feed additions. So they are also shifting the profiles here and there. So we are currently playing around in those areas. And having an understanding of that one, so it will give to the next step on how this would be modeled for the outcome that would be given by the adaptive model.
And another question was, I'm not sure if you have the answer to this, Abhijit, or not, but how accurate is the data compared to the traditional Glycan method?
Yes. So this is a topic about the data interoperability that is coming from all the different analytics we have. And this is a question that is quite well known in the analytical world, right? How do we bridge data that is coming from different analytics and so on? So that is where means what I highlighted here, that, okay, so let me show you that when we are going to perform the method adaptation and see how it is going to be performed. So we are going to compare it with the gold standard, which is the release glycan. And that is where means we are going to assess means how well, or let's say to what extent the level of information by this platform is giving in comparison to the reference method that is well established across.
Thank you for that. And I think we are up on time almost. So why don't we carry forward for closing statements? But I appreciate everybody adding the comments. And if you haven't got to your questions, we will get to them and respond to you. If there are any other questions, please do email to Jim so we can kind of take those into suggestions or recommendation. And if you'd like to be joining the program, please do reach out to Jim as well. I'll start up. Next slide, Abhijit. So how do we follow NIIMBL project? So this is just some administrative kind of closings here. You can basically log on to the NIIMBL website and you can select the program or the projects that you would like to follow. And it's pretty straightforward. It's basically, once you have a registration, you're registered at NIIMBL website, you get your own user ID and login, and you can basically just click on the follow button to get an updates on the project or the program of your interest. And you just have to click the updates for all the projects or all programs, and you automatically get an email as long as there's an update for what program established. Next slide, please. So I just want to remind everyone that we have an upcoming Big Data workshop on March 15th and 16th. The day one is open to all, so members and non-members of NIIMBL. So please take the advantage of this and join in and contribute to your ideas and to the solution and the roadmap that we are trying to create here. Day two would be open to members only. So we would like to join NIIMBL by that time. Please reach out to Jim and he can coordinate with you directly on how to enroll in the memberships. So this is a collaborative, sorry, can you go back? Sorry. So, we're seeking experts from biopharma manufacturing, both the technology developers, the CDMOs, and biomanufacturing themselves, to understand how we can simplify and optimize the lifecycle for biomanufacturing. And this is pertaining to big data roadmap development. Next slide. So in closing, I just want to remind the folks that, you will receive a survey link shortly after this webinar is complete. In an email, please complete the survey for an immediate access to the webinar recording. As soon as you complete the survey, a link will be sent out to you. The webinar recording will be sent out to you, so you can review those as per your leisure. I just want to remind folks that there are upcoming NIIMBL program webinars as well. The next one is next Wednesday on March 1st. That's where we have a case study for NIIMBL biomanufacturing process ontology development. That's been taking place within the Big Data work stream. So we'll have Big Data team update on the biomanufacturing process ontology on that aspects. And on March 8th, we have the NIIMBL vaccine manufacturing initiatives. So we'll get to hear from the work that's ongoing at that work stream. So you can register at NIIMBL.org at the bottom, and you'll get information for those. If you're not registered for those workshops, please go ahead and register for those. Is that the last slide? I'm not sure. Yep. All right. Thank you all for attending this webinar. It's been great help, and we look forward to basically seeing you in person or virtually during our workshop, And thank you, Abhijit, for taking this time and preparing to update the team members on the work that you guys have been doing.
Thanks a lot, and thanks a lot to all the participants on this interesting questions too.
Thank you.